everyone, welcome to my channel. In this video, we are going to see local and action potential from physiology. Before moving ahead, let's see the RMP values. Most neurons have RMP or resting membrane potential as minus 70 millivolts. Large motor neurons have minus 90 millivolts. Skeletal muscle fibers have minus 90 millivolts. And cardiac muscle cells also have minus 90 millivolts. Pacemaker cells in heart have minus 50 to minus 60 millivolts. Smooth muscle cells have minus 45 to minus 65 millivolts. Hair cells of cochlea, these are also having minus 65 millivolts as RMP. Rods and cones of retina, minus 40 millivolts as RMP. Epithelial cells minus 52 millivolts and RBCs is minus 10 millivolts. Now in this table, the given cells are actually excitable. However, epithelial cells and RBCs are non-excitable cells or non-self-excitable cells. Now, let's see the local potential first. Now we are considering most neurons, so we are taking RMP as minus 70 millivolts. Now let's say there's a subthreshold stimulus given at a point. What is a stimulus? Stimulus is any change in the external environment that elicits response from the excitable tissue. In this case, we have neurons as the excitable tissue. Now as soon as the stimulus is given, this RMP or the potential in the nerve increases to minus 65 millivolts and then it drops down. So this stimulus is not adequate enough to provide a action potential curve. So this is known as subthreshold stimulus. What is happening when stimulus is given? As soon as the stimulus is given, the voltage gated sodium channels open and then the sodium present outside the cell goes into the cell and then they close. And of course from minus 65 to coming back to the RMP, there is potassium channels opening. Or you have sodium potassium active pump which regulates the concentration gradient or maintains this concentration gradient and hence bring back the sodium into the ECF, causing the potential to drop down to RMP. Let's say I give another subthreshold stimulus and now this potential goes to minus 60 millivolts. But still it is not an adequate threshold potential for an action potential curve. So from minus 60 millivolts, again as the sodium channels will close, the potassium channels will open or sodium potassium ATPase pump will be active and these will cause the potential drop down to RMP again, that is minus 70. So local potential is caused by the subthreshold stimulus. Let's see the action potential. Now let's see again I have the RMP as minus 70 millivolts and there's a stimulus given. The potential rises to minus 55 millivolts this time. Now minus 55 millivolts is a threshold potential or firing potential in most of the neurons. So as soon as the potential reaches minus 55 millivolts, these sodium channels remain open and therefore the potential further increases to 0 millivolts and causing the potential to reach in the positive voltage, and that is going to plus 30 millivolts. At this plus 30 millivolts, the sodium channels, voltage gated sodium channels close and potassium channels open, which lead to repolarization or drop down of the potential from plus 30 millivolts to minus 70 millivolts down. So what we say is the stimulus given which leads to action potential is a threshold stimulus. Let's see the difference between local and action potential. Local potential occurs when we give subthreshold stimulus, and action potential occurs when we give threshold or suprathreshold stimulus. Local potential, there are graded responses, whereas action potential is all or none responses. What does this mean? This means that any subthreshold stimulus will give a response in local potential. There will be increase in the potential and then again drop down. But in action potential, if the stimulus is not adequate enough so that the potential reaches the threshold potential, there will be no response at all. But once the potential reaches the threshold or firing potential based on the stimulus strength, then there will be a full response. Next point is local potential is have decremental changes. That is, it decreases over time and space. Whereas action potential travels without decrement. There is no change in amplitude. Local potential is not self-propagated, whereas action potential is self-propagated. Local potential may or may not be followed by some action, but action potential is always followed by some action. Like, let's say there is secretion of some glands or there is muscle contraction. Local potential can be depolarizing or hyperpolarizing. Depolarizing, that is EPSP, excitatory postsynaptic potential, and hyperpolarizing, it is IPSP, inhibitory postsynaptic potential. You will be learning about them in a bit detail in synapse part in CNS unit. Action potential is always depolarizing. Local potential can be summated, whereas action potential cannot be summated. Now there are two types of summation, spatial and temporal. Let's say this is a neuron cell body and I have given stimulus at these areas. Now all these areas stimulus gets added up and then they produce a local potential. In temporal, let's say I have selected one area and I am giving stimulus more than one time at this same area. Then as the frequency of subthreshold stimulus increases, I get the local potential, which is summated response. Example of local potential is EPSP, IPSP, motor end plate potential present at the neuromuscular junction, we know that. Then receptor potential. You'll be studying receptor potential again in the CNS unit. Now let's see the action potential in detail. Let's see their phases. We have RMP as minus 70 millivolts. Now as soon as we give a threshold stimulus, this potential increases to the threshold potential or firing potential, that is minus 55 millivolts. And after that it goes on increasing, crosses 0, and then there is an overshoot above the 0 millivolts towards the positive voltage, and there is the spike formed from 0 to 30 millivolts. Then there is decrease in the potential going down to the RMP, but then it still decreases below the RMP to minus 71 or minus 72 millivolts, and then there is slow increase back to the RMP. Now the first one, where the RMP value is minus 70 goes to plus 30 millivolts, that phase is depolarization phase. From plus 30 millivolts to till the voltage goes to the RMP or below to the RMP, we say it repolarization phase. Now when it reaches the RMP, we say it after depolarization, and after the depolarization we have another phase, that is after hyperpolarization. Now when the potential changes from -71 or -72 back to RMP, that phase is after hyperpolarization. Now, this 0 to 30 we have overshoot. Now, what is this after depolarization, which is there, and there is repolarization in between depolarization and after depolarization? Why is it named as after depolarization? This is named on the sequence of discovery. That is, after depolarization phase discovery, we have discovered this after depolarization phase, and then we have discovered the repolarization. So the name is just controversial name, but yes. Now let's see the causes of depolarization. Depolarization is due to Na plus influx because there is opening of sodium voltage gated channels which open at the firing level or -55 millivolt level. Repolarization is due to closure of the sodium channels and opening of slow voltage gated potassium channels. After depolarization, now what is this actually? There is accumulation of K plus outside the gates. Then there is efflux of K plus reduces slightly. Because of this, the curve becomes less steep as considered to the initial repolarization. And that phase is called as after depolarization. Then we have after hyperpolarization. Now K plus or potassium voltage gated channel close slowly as well as open slow. Let's see here the closing slow part. So because of this, the K plus efflux occurs beyond the RMP. This leads to hyperpolarization. Now sodium potassium ATPase pump is required to maintain the concentration gradient, and this pump brings back the K plus inside, and hence the potassium becomes less negative, that is towards RMP. Now let's see these voltage gated sodium channels and voltage gated potassium channels. Only one stimulus is required to activate both the channels. But why is it that the sodium is acting in the depolarization phase whereas potassium is acting slowly later on in the repolarization phase? This is because voltage gated sodium channels are fast channels. They open first, whereas voltage gated potassium channels are slow channels and they open later. But only one stimulus is required to stimulate both the channels. Now we can see that voltage gated sodium and potassium channel both are open in voltage range of -70 to +30 millivolts, which is same for depolarization and repolarization. However, as K plus is slow, it opens later on. Voltage gated sodium channel is fast to close also. As it is fast to close, the depolarization is quickly followed by repolarization. Whereas voltage gated potassium channels are slow to close, and because of this there is hyperpolarization after repolarization. Now the sodium channels have two gates, inactivation and activation, whereas potassium channel has only one gate, which is towards the ECF. Let's see these sodium channels gates in detail. Now you have one gate, that is activation gate towards the ECF, and an inactivation gate towards the ICF, which is actually opening by ball and chain mechanism. Now at rest, the activation gate is closed and inactivation gate is open. But as one of the gate is closed, the sodium entry or exit will be 0 and hence no change in the potential. When a stimulus is given, now the activation gate opens, and now we can see that inactivation gate was already open. Now activation gate has opened, so the channel can now leak the sodium into the channel, into the cell. So this is activated state of the sodium channel. As soon as the potential reaches plus 30 millivolts, the activation gate is open, but this inactivation gate is closed first. And this is the inactivated state. Just remember that inactivated state, when the potential reaches plus 30 millivolts, the inactivation gate is closed first. Then in mid or late repolarization, we get the resting state back, where the inactivation gate is again open but activation gate is closed. Now this inactivated state, where the inactivation gate is closed, this is responsible for refractory period. Let's discuss this refractory period also. Refractory period is a duration during which second stimulus cannot produce response. Now there are two types, absolute and relative. In absolute refractory period, no response is produced irrespective of the strength of the second stimulus. This absolute refractory period is from the firing level, that is minus 55 millivolts level in the depolarization phase, till the 1/3 of the repolarization phase is complete. Because sodium channel is in inactivated state at that time. In relative refractory period, we have only response for supra-threshold or stronger stimulus than the threshold stimulus. Now this relative period is from 1/3 repolarization phase till the start of after depolarization. In the after depolarization, the excitability is increased slightly. So here we have absolute refractory period and this is relative refractory period. Now let's see the last part of the action potential, that is the site of generation of action potential in the neuron. Now this is your cell body. This is somadendritic zone where you have graded electrogenesis or you have local potentials, and this is the axon. Now this portion is called as axon hillock. It is the dilated part of the cell body itself, which from where the axon arises. So axon hillock is a part of cell body, but axon arises from there. Initial segment is the part of axon, initial part of axon that is unmyelinated. Okay. Now in these both areas, axon hillock and initial segment, there are very high concentration of sodium channels per unit area. So the site of generation of action potential in spinal motor neuron, it will be initial segment more than the axon hillock, because initial segment has more sodium channels as compared to the axon hillock. So site of generation, if they ask you a question in spinal motor neuron, and if they give you options and there is two options, initial segment and axon hillock, your answer should be initial segment. But if initial segment is not given, then your answer will be axon hillock. If axon hillock is not given, then your answer will be initial segment. Now, the site of generation of action potential in sensory neuron is different. In sensory neuron, the site of generation of action potential is initial or first node of Ranvier. Please remember this. In spinal motor neuron, the site of generation of action potential is initial segment and axon hillock. But in sensory neuron, the site of generation of action potential is initial or first node of Ranvier. That's it for action potential. Please like and share this video. Thank you and subscribe my channel.